Okay, so our final speaker of this session is Alexander Borowsky from University of California, Riverside, where he's a NIFA pre-doctoral fellow in the lab of Julia Bailey-Serres. And today he will be talking to us about the gene network discovery and engineering to enhance rice roots resilience. Your slides are up. I think I can hear you. Take it away.
Great. Hi, everyone. Kaisa, thank you for the introduction. And thanks to the organizers for having me for this talk. So I'm going to speak today about our work in gene network discovery and engineering to enhance rice root resilience. So rice is cultivated in these dynamic multifactorial ecosystems, from deep water environments to paddies, as well as upland irrigated environments. And the rice grown in both paddies and in upland irrigated conditions can be subjected to drought, and paddy rice can also be subjected to periods of complete submergence. And depending on the season, that period of submergence might be followed by a period of drought. And so the extreme weather events that cause these environmental extremes can also be exacerbated by climate change. So there's more frequent droughts and more frequent floods that farmers have to contend with. Additionally, paddy rice is a problem because of methane emissions from the bacteria in the anaerobic conditions. And so one potential solution for that is periods of dry down. And so that also causes some potential problems for the rice varieties that are cultivated there. And so we're looking at the root system, particularly because it's the plant's primary interface with water availability. The rice root consists of multiple cell layers, just like we heard about with the from the previous speakers. And it includes some cell types that Arabidopsis doesn't have, including this layer, which Kaisa mentioned, the exodermis. And so this is a barrier cell layer that serves specialized functions. One of those functions that we are interested in is this formation of a suberin barrier. So suberin is a waxy polymer. It's well known for making up the dry mass of cork. And so it has a function that we believe in different conditions to provide a barrier and protection. So if we stain for suberin using floral yellow, in rice, we can see that under water deficit stress, there's a big increase that's restricted mostly to the exodermis. We think this is helping to retain water. And we also see a similar thing happen in plants that are grown in a flooded environment. And in that case, we think that it's a barrier to help retain oxygen and help funnel oxygen to the root tip. So this is an interesting trait for us because it is potentially important for resilience in multiple environments. In the Bailey-Serres lab, we've leveraged not yet single cell sequencing, but other tools to understand gene activity in plants that I think have some potential advantages actually over the newer single cell approaches that we've heard about today. The first being translating ribosome affinity purification, or TRAP-seq, where we have a GFP flag tagged ribosomal subunit protein, which enables us to immunopurify ribosomes and their associated mRNAs. And then we can sequence those mRNAs and get what we call a translatome. So these are mRNAs that are being translated into protein. The other technology is that we use is similar, which is called Intact, allows us to purify nuclei from specific cell types as well. And then we often follow up our nuclei purification with ATAC-seq, so assay for transposase accessible chromatin. And this is a method that uses a transposase which inserts preferentially in accessible chromatin regions to identify regions of accessibility that are generally broadly important for the regulation of genes that are nearby those accessible regions. These technologies enable profiling of specific cell populations. So here I'm showing you one of our trap lines, which is particularly important to what I'm working on, which is marked by the LSI1 promoter. And this marks the exodermis and the endodermis in rice. This is marking the differentiated cells, which are potentially harder to access by single cell methods. The other advantage that these methods have is that they enable profiling of gene activity across multiple environments. These methods are compatible with flash frozen tissue, which means that we can subject plants to different environments and capture their translatome or accessible chromatin at a given state or at a given time. It's also, these methods end up being cheaper once we've got this collection of trap and intact lines. So we can profile quite a bit of different conditions, which is what Mauricio and Germain did here. And I'm showing a lot of conditions, and all this data is in our paper from this year. But today I'm going to talk specifically about what we've found with respect to water deficit. And so we profiled a number of different cell populations with these different promoters for TRAP, including the LSI1 promoter, which I mentioned. And that allows us to define patterns of the different translatomes we find. So here on the x-axis, what I'm showing is the different promoters used to drive TRAP, as well as the different conditions where these lines were profiled. And then on the y-axis, we have genes that are grouped by having a shared pattern in the translatomes. And then the heat map is filled with the relative abundance of those RNAs. So one thing that we can see first is that the LSI1 translatome has quite a lot of distinctions from the translatomes of the other cell populations. But we can also see things where it's specific not to a cell type, but specific to the response. So this group of genes here, we see as low in control conditions, high under water deficit conditions, and then low again under recovery conditions. So this is quite specific to the drought stress, and it's common across all cell types. And this includes genes that are canonically associated with ABA response. We also see responses that are more specific. So for example, here we see a set of genes that is at first enriched in the LSI1 translatome, but is then downregulated by water deficit. And this includes a lot of genes that are involved in metal ion transport and homeostasis, particularly iron. And then finally, we see that this group of genes here is enriched in the LSI1 translatome, but upregulated by water deficit. And this includes genes that are involved in suberin biosynthesis, which is what I was mentioning earlier. We are also interested in what's regulating these sets of genes. And so we can look for enrichment of transcription factor binding sites in the promoter regions, or not the promoters, but the accessible regions that are proximal to these genes. And this identifies, for example, for the ABA response genes, the BZIP transcription factor binding sites are particularly enriched, which is what we'd expect, given that the ABA responsive transcription factors are in the BZIP family. And in suberin, we see enrichment of MIB transcription factor binding site, which is consistent with the known regulators of suberin biosynthesis. We can further refine with co-expression a set of genes that are responsible for the biosynthesis of suberin in the rice root in response to drought. And so this includes everything from the regulation to the synthesis of the individual parts, and then export and polymerization at the cell wall. We can then also look for the transcription factors that are here and are enriched, and map the transcription factors, which are the empty circles here, to potential target genes, which are the filled circles. So with this information, our next question is, how can we use it to manipulate this suberin? And so to do that, I've taken sort of two different approaches. First, to see if we can decrease the suberin by CRISPR-Cas9 mutagenesis of some of these targets. as well as we also want to be able to increase the suberin. And so we're using a sort of a transcription factor misexpression approach for that. So far, I've been able to demonstrate that we can disrupt some of these rice genes with CRISPR-Cas9 and reduce suberin by floral yellow staining. So I'm showing you a wild type rice plant as well as 3 different single gene knockouts, which are grown on plates and treated with ABA and stained with floral yellow for suberin. And we can see a reduction in the stain intensity in the mutants. And so I'm excited to have these genetic resources to be able to evaluate the role of suberin in some of the stress responses that we're interested in. Additionally, we have a lot of potential transcription factors that may regulate suberin based on our network analysis. And to express them all in rice with different promoters driving them would require a lot of transgenics, which would take a long time and be quite expensive for rice. So I'm first trying to prioritize what we might use for engineering with transient assays and bentamiana leaves. So we've cloned transcription factors from our suberin network and fused them to a 35S promoter and an orange fluorescent protein. As well as also we've cloned promoters of some of the enzymes in the suberin network and fused those to a GFP GUS reporter. If we co-infiltrate these 2 plasmids together and measure the ratio of GFP to MKO kappa on a plate reader, we can get an initial readout of how capable a given transcription factor is of activating a given promoter. And so we've looked at these across different, we've across 3 different promoters, and I'm showing here 5 different transcription factors and a control, which is just a nuclear localized fluorescent protein. And we can see that the MIB genes that we've used seem to be the most potent activators of these promoters, which is not at all surprising. But they may not be fully functionally redundant. We see some variation depending on which MIB we're looking at. We're also leveraging the trap attack-seq data that I mentioned earlier to select promoters to drive these transcription factors in rice so that they're active in the cell types and the conditions that we're interested in studying. And so with that, I will acknowledge our collaborators on this work, which includes 2 NSF PGRP projects, the Plasticity Project, the Secretome Project, the Bailey Sayers Lab, and the people that I've worked with there, as well as the NSF, the Plant 3D program at UCR, and the USDA NIFA for funding. Thank you.
Thank you, Alex, for an excellent talk. There's no questions at the Q&A yet, so I'm gonna start again with my questions. So when you showed that heat map of the various responses and how different cell types respond, I noticed that there's actually quite a lot of clusters. So it looks like that all of the 13 to 20 are more or less the barrier cell type specific or enriched responses. So what else is there than the suberin and the iron responses?
That's a good question. I don't remember off the top of my head what exactly the genes are in those groups. The things that we do see in this cell population, which I think are really interesting, are we see an enrichment of genes involved in nitrogen assimilation. So I think that that's a potential role that the exodermis, I believe, plays, is that it has this role in nitrogen. We also see that there's just sort of a lot of transcription factors that are enriched here. It's something, it seems like the transcriptional profile of the cell type is very tightly controlled and regulated.
Cool. And then a question about the transcription factor transients that you're doing in bentamiana. So you're actually seeing MIB transcription factors able to work alone to induce expression. So we might have been trying similar sorts of things a little bit. And one of the hypotheses that we have is that they might need to have a dimerization partner.
Yeah, so
So,
I think that the benthammiana assay isn't perfect because it's quite possible that we are activating endogenous benthammiana suberin regulators. So that's sort of it's possible that they're recruiting something in benthammiana that we don't know about. I also know that at least in the area of the phenylpropanoid regulation, there are some MIBs that require partners and there's some that don't. And so it's possible that depending on which transcription factor you're looking at, you may or may not need other partners.
all right, we have some Q&A questions coming in. So Josh Cohn is asking, have you considered yeast-1 hybrid to identify transcription factor binding to regulatory sequences identified in genes of interest? And also you mentioned not promoters, but clarified proximal regions. Were those regions identified by ATAC-seq, and how far are these from genes of interest?
Yeah, so the first question about yeast-1 hybrid, I think yeast-1 hybrid is a great platform for this sort of experiment. We didn't do that here in part just because we had already sort of started cloning things for going into plants. And also because the extent of activation is what we're interested in, and the yeast-1 hybrid system uses a consistent activation domain regardless of what transcription factor you're studying. So the magnitude of activation, I think, is something that we can potentially get here that we couldn't get from the yeast-1 hybrid. Yes, I clarified proximal regions. So those are regions that were identified by ATAC-seq, and they vary based on how far they are from the gene of interest. But there's I can't remember off the top of my head how far it is, but it can be up to each accessible region is assigned to its nearest gene. So it could be 5 kb upstream. If that's the nearest gene, then we have an association.
Great. And then from David Jackson, why do you think iron is specifically regulated and why not other metal ions?
So I do think we see some things. So like there's some metal ions that are sort of redundantly regulated with iron, including like manganese. And so we do see some of those things as well. I think that we're not quite, so there's a few different ideas we have going on here. One I mentioned is that that nitrogen assimilation is enriched in this cell type. And there's a few enzymes in that pathway that require an iron cofactor, nitrite reductase, and ferredoxin-dependent GOGAT. The other thing is that this is a barrier layer, and so it could be facilitating transport. But it really does look to me like the cell population has a demand for iron that's localized, at least by the translatome. So that's sort of what we're thinking about. Hopefully we'll be able to also do some genetics there as well to try to tease out what's going on.
Excellent. Thank you. Thank you, Alex. Thank you to actually all of the speakers of this session.